All right, welcome everybody back to Statistics 115. Today we're going to talk about epigenetics. So in the last lecture, if you remember, we talked about ChIP-seq, which you can map genome-wide transcription factor binding sites. When we did ChIP-seq in the human genome, we found that if you remember, the transcription factor is able to find its binding site because of the presence of a sequence pattern that it recognizes. So you would imagine when a sequence pattern happens, the TF will go there and bind. But once you do ChIP-seq, you realize that in the genome, there are many more times this motif appears, but the binding site in that particular cell is only a small percentage of all the occurrences of that motif. So the question is, how come not all the motif sites are bound by this factor? In addition, if we were to do the ChIP-seq for the same transcription factor in different cells, for example, for estrogen receptor, it's a transcription factor. It's known to bind, it's at least expression level-wise, we know that it's expressed in the pituitary for puberty, the sex differentiation. It's expressed in the breast, in the ovaries, in the uterus, or in and also it's expressed in the bone for female, like bone density. It's expressed in the liver. But if you were to do a ChIP-seq of the same transcription factor in many different cells, you would find that their binding sites only have small percentage of overlap. Usually a quarter of the binding sites overlap or a half of the binding sites overlap. So very often majority of the binding sites do not overlap between different cells. If the TF is recognizing the DNA sequence to bind, why do they go to different locations in different cells, right? And also, how come when the TF is binding to a particular, in the cell state, it's binding to tens of thousands of locations, but only regulate a subset of the nearby genes? And because of all of these reasons, people are more and more interested in epigenetics, and that's how I got drew into epigenetics. So in some sense, for me, from 1997 to about 10 years, we worked on motif finding. But we knew motif finding was not going to really solve the problem. So starting from about 2001, we worked on ChIP-ChIP, then later on ChIP-seq for another 10 or so years. And once we have ChIP-seq or ChIP-ChIP done in many different cells, starting about 2007, 2008, we, as well as many other people, started interested in epigenetics when they see, wow, the same TF go to very different locations. And so we started learning what does it mean by epigenetics. We mentioned briefly in the last lecture, the DNA is a double-stranded sequence. If you were to just stretch it out, if you just read out the nucleotide of 3 billion base, you get the DNA. It's actually a very, very long, tiny sequence. But in order to package this thing into the cell, there are many different levels of packaging. You can see initially this is the DNA. It first packaged into something called a nucleosome. A nucleosome is the basic unit of the chromatin. The chromatin basically is you're talking about the structure of the DNA, and not just on the double helix, but higher like structures. So nucleosome here, you can see here, it's 146 or 147, so roughly 150 base pair DNA that's wrapped around some histones in here. And but if you were to zoom out a little bit, you will see this, and you can think of this as pearls on a necklace, or you can think of as some strings and Oreo cookies. And so each of the Oreo cookie is about 150 nucleotide, and then there are some space on the DNA like link them together. And then if you continue to zoom out, you will see this thing. They kind of keep, it's like a rubber band. You keep twisting, and they form even more and more structures, right? So this is, you can see all the blues are Oreo cookies stacked on top of each other. And then, if you keep on zooming out, you will see this thing. And this is what people call the 30 nanometer fiber. That's what most people call as the kind of chromatin. They consider this fiber. And this continue to, if you continue to zoom out, you will see that they go back and forth and back and forth and back and forth. And you continue to zoom out, you will see this like this. And then they, yeah, like this. And eventually, when the DNA is being replicated during cell cycle, you will see that these are the two sister chromatids. So they are initially from the same chromosome, but the DNA replication duplicated the DNA. And you can see the chromosome in its highly compacted structure. In reality, that's like a lot of these, it's like a rubber band keep on squeezing, and they just twist and turn. Then they form this really very high level chromatin structure, okay? And so in the cell, in our body, every cell is in a different shape or different state. For example, the cells that are dividing, their chromosome is in a really compact structure that you can see these eggs. But the cells that are quiescent or they are kind of stable or doing its regular function, you might see more kind of open structures. And also within this chromosome, there are also some regions that are more open, but other regions that are more closed. And so this is really epigenetics. You can see it because the DNA is the same. Every cell have the same DNA, but they have very different state. And so epigenetics official definition is functionally relevant changes to the genome or that do not involve a change in the nucleotide sequence. So when we talk about epigenetics, there are two levels. The first is the study of heritable or transgenerational changes in gene activity that are not caused by changes in the DNA sequence. For example, I will show you some examples later. From one generation to the next generation, there are things that are inherited, but is not coming from DNA. This is transgenerational change, but also people who are interested in epigenetic in recent years, they also want to study stable long-term alterations in transcriptional potential of a cell that are not necessarily heritable. So let's talk about some this either transgenerational or upbringing. You know, like there's the nature and nurture. The nature is the DNA part, and nurture is, you can see the nurture part. So in a beehive, the queen bee will give birth to many little babies, and they are kind of raised in this their house, beehive. And each of this well has a larva. And depending on what they are fed, if they are fed with regular food, they grow up as a worker. But if they are fed with royal jelly, they can have the potential to become a queen. And the queen, but once these little, new little queens grow up, they have to fly out and establish their own new colonies, right? Then they can survive. In fact, there are some of the ants have kind of similar situation. Sometimes if you take out the queen ant, there will be several other stronger ones. Their morphology will change and they will fight to become the new queen. And then that new queen will continue to maintain the remaining colony. And so you can see whether they are a queen or worker, their looks are totally different, their behavior, right? Everything is different, but genetically they are kind of the same. So this is an example of epigenetics. Another case of epigenetics is discovered in the mouse. So when the mother mice are pregnant, depending on whether they have methionine-rich diet versus methionine-poor diet, you can feed the mice with a different diet, one rich in methionine. When you have methionine, the DNA on certain locations will get methylated because you have the methyl group. Then the baby mice will come out looking thin and healthy like this. However, if the pregnant mother mice are fed with methionine-poor diet, they will give rise to babies which, when they grow up, will have this yellow hair color as well as obese phenotype. And they are called Agouti gene. So this is one gene that controls hair color and also metabolism. And so this gene, when it's not methylated, the mice will have yellow hair and obese phenotype. And so you can imagine this is definitely not happening directly on the DNA sequence because the mom has already had the baby in the womb. The DNA is already set. But because of nurture, in some sense, because they are raised in different ways, then you'll have a different outcome. This is another example of epigenetics. This is a zygotic twin, which means their DNA is the same, but you can see they kind of look different. They have different characteristics. For example, you can imagine with two twins in the mom's womb, they are kind of competing for nutrients from the mom. And sometimes one baby is weaker, the other is stronger. Their personalities, their temperament are very different. And some of that is coming from the epigenetics. In fact, there is one study showing in 1998, there is a big ice storm in Northern America, in Canada. For a week, they lost the power and it was quite chaotic. And many years later, there are epidemiology studies showing that the kids that were, well, the fetus at that time, later on the mom had their regular births, the kids were growing up. These kids actually are going through a very tough childhood. They're kind of in all sorts of troubles and not very, or like behavior problems because their mom were under so much stress during this pregnancy period when, there's ice storm. And so the stress situation can influence the child behavior. In fact, during World War II, there was a siege in the Netherlands. So German soldiers had a siege in that region. A lot of people died, but then some people survived even with the famine. And it was found several generations after, their kids are more likely to be obese. And so it's hard to imagine this is genetic selection, but probably epigenetics. Something happened if they have this really extreme fasting experience, then their next generation is more likely. I guess they have learned to adapt and accumulate nutrients when there is nutrients available. And the result is that these kids, or even third generation, might be more obese. Okay, so you can see epigenetics change things. So that's on the transgenerational. Well, so some of this is transgenerational, but some of this is just the nature versus nurture. But on the cell level, you can imagine when we have an embryonic stem cell, it already determines the DNA. At that level, it has the potential to differentiate into different cells, for example, muscle or liver or bone or skin or whatever, gut, all the different cell types. The DNA is the same, but then it has the potential to differentiate. But for example, if the differentiation already picked one path to say, now this lineage is going to develop into blood, then it can only be differentiating into like red blood cells or white blood cells. It's not going to be differentiating now into a hair or a skin, right? So every cell knows its identity. And when it replicates itself, like in our body, all of our cells replenish, they divide, they make new cells, old cells die. How does each cell know that, say, if I'm a heart muscle, the next cell I'm going to make is still a heart muscle instead of a liver, right? That is also epigenetic. Every cell remembers its state. And in order to maintain that state, you need epigenetics. So in the early 20th century, Waddington proposed this epigenetic landscape, right? when the cell is a pluripotent embryonic stem cell, it has a lot of potential. You can think about the actual transcription events happening as the kinetic energy, but the epigenetics as the potential energy. And so when you are embryonic stem cell, you are on a high epigenetic potential, but as you differentiate already, they kind of go down the hill, and they will just know how to go down, harder to go up. Although this is really changing in recent years, people are doing IPS, which is induced pluripotency. For example, you take a piece of skin cell, which is already differentiating towards the end, and you add a few factors, and then now they become an embryonic stem cell. You can create a whole animal from this one thing. You can clone a sheep this way. And that's because of a reprogramming on the epigenetics. Okay? So that's epigenetics. Questions?